So this is our lab report, Lab 6 on metabolic tests, Group 8 by Viviana Soto and Kaytlyn Torres. So hi everyone, today our group's lab focus on metabolic tests, basically how different bacteria use or break down carbohydrates and what metabolic products they produce. The main objective was to observe how these bacteria generate acids or gases through carbohydrate utilization and interpret those reactions to help identify unknown organisms. The purpose behind these tests is to understand how microorganisms obtain energy either through oxidation or fermentation of sugars. This helps us tell apart oxidative, fermentative, and non saccharolytic bacteria, which is really important for identification. So, one question we considered here was what is the purpose of performing metabolic tests on bacteria? Another one was why is it important to include control tubes in each test? So basically, the controls make sure that any color or gas change actually comes from bacterial metabolism, not just a random pH change or contamination that might have been included. And in our background, we mentioned some tests we use like OF glucose, TSIA, and phenol red carbohydrate broths. These are common in diagnostic microbiology because they tell us a lot how bacterial enzymes activity and sugar metabolism. So for this experiment, we used a variety of bacterial cultures, including E. coli, Salmonella, tr- Triotherm, Strigolflenexneri, Protinus mirabilis, and the unknowns, which were 118 and 298. Our media included the OF glucose tubes, the triple sugar ionic or TSIA, and the phenol red carbohydrate broths for glucose, and then lactose and sucrose. And the equipment was pretty standard. We used like the loops, the needles, Bunsen burners, the mineral oil, the incubator, and the labeling tape. So for this point of the lab, we asked what is the role of mineral oil in the closed tube, where sugar are present in TSIA, and why are they included? They're key, the mineral oil, because it creates an anaerobic condition for fermentation testing, while other different sugars help identify specific metabolic activities of each organism. So for the OF glucose medium, we used two tubes per bacterium. One labeled open, the other one was closed with mineral oil added. The open test was for oxidation since the oxygen is available, and the closed one is for fermentation since the oxygen is blocked. Here, what is the purpose of using both open and closed tubes, and what indicator is used in the OF medium, and how does it work? So the indicator is the bromoethyl blue. It turns yellow when acid is produced, meaning glucose metabolism occurred. If only the open tube turns yellow, that means oxidative metabolism. If both turn yellow, then fermentative metabolism. And if neither change, then that suggests there's no carbohydrate metabolism at all. Then for the TSIA, we stab and streak the slant with our bacteria. This then led us to discuss like what does the red slant and yellow butt mean, and what does a black precipitate indicate. These results tell us which sugars were fermented, which were glucose, lactose, or sucrose, and if the hydrogen sulfide gas was produced. Cracks or bubbles also mean gas formation. For the phenol red carbohydrate boss, we inoculated tubes containing glucose, lactose, and sucrose. We made sure to not disturb the small tube inside the durum. That's where the gas collects during fermentation. At this step, we mentioned like what's the purpose of the durum tube and why shouldn't we disturb it? It's because like the tube traps gas bubbles and shaking it can release them, giving false results. What does the yellow color mean? Acid production, positive fermentation, and the red or pink color would indicate like no fermentation or alkaline reaction. This test also helped us talk about how can we tell strong fermentators from weak fermentators. The strong ones change color faster and often form gas. So here are some of our results. This was the sucrose broth, and you could see the unknown and the control. Um, we could see the unknown 118, our E. coli, or unknown 298, and our P. aeruginosa. And then here are the other ones. This is the lactose broth. This is the glucose broth. You can see a slight difference in the yellow as one's more clouded than the other ones. The TSIA, and it continues. And you can see how we got a control for each one. You see we can control the different colors and how they looked different from the control. So here we show the results. Well, there's we're showing the results from the different tests. Each photo compares how each bacterium related to different media. The color really helped us figure out the metabolism types. Um, so like I said, that what does the yellow in both OF tubes indicate, or what does no color change mean? For TSIA, we refer to what does yellow slant in yellow butt mean. That shows fermentation of glucose plus lactulose or sucrose, and the black precipitate means that indicates hydrogen sulfide reacting with iron salts. And then we saw, um, if like when we saw cracks or lifted agar, like how does that cracking occur? And it's due to like the gas produced during fermentation. So in conclusion, these tests really helped us identify how each organism metabolizes sugars. The OF glucose test separated oxidative from fermentative bacteria. For example, the A. bacillus and the P. aeruginosa were oxidative non-fermentators, while the E. coli and the unknown 298 fermentated glucose. TSIA gave us more detail by showing which sugars were fermented. We saw yellow we saw the yellow for the strong fermentators and the red yellow for glucose only fermentators. The phenol red broths helped us confirm acid and gas production. Again, E. coli and C. frundi turned yellow with gas bubbles, while A. bacillus stayed red. So throughout the experiment, we mentioned questions like what does gas production look like in TSIA? Why is it important to include controls in each set of tests? And how can you tell strong versus weak fermentators? So those questions helped us realize how important these metabolic tests are in identifying unknown bacteria and confirming their energy pathways. Thank you.